Exposure to intense radiation kills most life in seconds. DNA damaged, proteins destroyed, cells reduced to debris. But as usual, life finds a way. Because there's one organism that can survive doses of radiation thousands of times higher than that which would kill a human. It can survive complete dehydration. It can even survive in a vacuum. It's called Deinococcus radiodurans, nicknamed Conan the Bacterium. And understanding how it manages to accomplish this could tell us what the true limits of life really are, if they exist at all. Now you might think that Deinococcus would have been discovered in a nuclear reactor or a radioactive wasteland. And you'd be wrong. It was actually discovered in the 50s by scientists who were experimenting on new ways to sterilize canned meat using gamma radiation. See, these scientists took tinned meat products and exposed them to huge doses of gamma radiation, which should have been enough to kill anything inside that could spoil the meat product. But the meat still went bad. When these scientists looked closer, they managed to isolate a bacterium that had not just survived the radiation, but was actually growing quite normally. Needless to say, this was unexpected. Now, the numbers on how tough Deinococcus really is are, quite honestly, absurd. It can survive up to 5,000 grays of ionizing radiation with no loss of cell viability. Everything survives. It can survive up to 12,000 grays with loss of viability of 90% of cells. And that might sound like a lot, but that still leaves 10% of the cells to regrow as normal. And that's more than enough for a bacteria. To put these numbers into context, around 5 grays is enough to kill a human, and 4,000 is enough to kill another famous survivor, the tardigrade. But it's not just ionizing radiation. Deinococcus can also survive more than 500 joules per meter squared of UVC radiation. That's 33 times more than E. coli can survive. It can also survive for years, completely desiccated, dried out. It can survive oxidative stress, freezing, and even in a vacuum. But how is it able to survive this? Well, radiation mainly kills cells by causing damage to the DNA. Ionizing rays, like gamma rays, fragment chromosomes into smaller pieces. And if the DNA ends up too fragmented, then the cell isn't able to repair it and ends up dying. But Deinococcus does something extraordinary. After exposure to extreme radiation, which leaves its genome fragmented into dozens or hundreds of pieces, it doesn't panic. It meticulously reassembles its genome back together the way it was. And it does this by using overlapping fragments. Deinococcus is able to reassemble its genome almost like a molecular jigsaw puzzle. See, fragments with matching sequences are annealed to one another in order to progressively extend the intact region of genome using single-stranded annealing. The complementary regions of these sequences are then filled in, and double-strand breaks are repaired using homologous recombination. Through this system, Deinococcus can reassemble its entire genome in a matter of hours. The process is called extended synthesis-dependent strand annealing, and it's one of the most efficient DNA repair systems known to science. Now, for a long time, scientists thought that the DNA repair system explained Deinococcus's extreme resistance to radiation. But it turns out that that's only part of the story, because gamma radiation does actually also damage proteins, including the enzymes responsible for protecting DNA and repairing it. And so Deinococcus has evolved mechanisms to protect those proteins as well. It does this by accumulating high levels of manganese ions and small molecules that act as antioxidants. These protect proteins from the oxidative damage that radiation exposure causes, and means that they're still functional after the radiation exposure is finished. So it's not just a case of Deinococcus repairing its DNA more efficiently, it also protects the repair system better. But Deinococcus's unique cell architecture also plays into its ability to resist radiation damage. See, Deinococcus typically grows in tetrads, as groups of 4 cells that cluster together. It also maintains multiple copies of its genome in each cell, and it clusters these different copies of the genome into a small space so that they're all next to each other. What this means is that Deinococcus has redundancy, so that when its DNA is damaged, it's more likely to be able to find fragments that overlap with one another to assist in the repair process. On top of that, by keeping its genomes clustered together within the cell, it speeds up the process of repair because they're all located in the same region. It doesn't have to go all throughout the cell to try and find these fragments. On top of this, Deinococcus is able to take up DNA from outside its cell. So, in a tetrad, if one of the cells dies as a result of radiation damage, it releases its genome into the environment. The other 3 cells can then take up that DNA and use it to help reassemble their own genomes, improving their chances of survival. It's an entire system built around survival. Now, despite its amazing abilities, Deinococcus doesn't, as you might think, live in environments that have extreme exposure to radiation. It's actually often found in things like dry soils, desert environments, dust particles, or places with high sunlight exposure and dehydration. And so scientists at the moment believe that its ability to resist intense radiation is actually a side effect of the mechanisms that it evolved to deal with its real challenge, desiccation. Because drying out a cell causes damage to DNA in a very similar way to what radiation does. So if you can survive one of them, you can probably survive the other. Now, aside from the obvious fundamental interest in understanding how Deinococcus does what it does, it also has some potential real-world applications. For example, it's being explored for its uses in bioremediation. Can we potentially engineer microbes to help us clean up radioactive waste and sites exposed to nuclear activity? It's also helping us understand DNA repair pathways, which is relevant particularly in things like cancer research. And it's aiding in the design of radiation-resistant enzymes for application in things from biotech to medicine. It's a model organism for understanding how cells can recover from extreme damage. It is also, though, raising some very interesting fundamental questions about what life can survive. If there are organisms on our planet that can survive these kind of conditions, what does that mean for potential life elsewhere in the universe? Deinococcus has survived experiments involving vacuum exposure, radiation consistent with space, and long-term desiccation. It raises some serious questions about the potential for microbes to survive through space travel and the resilience of life in general to space environments. In short, life may be a lot tougher than we once thought. Deinococcus is also interesting because it doesn't avoid disaster. It embraces it. Rather than trying to evade damage, it specializes in being able to recover from near total destruction. Its existence reminds us that evolution is about survival. And sometimes it's not about how well you can avoid danger, but how much damage you can take and rebuild yourself from. Well, that's it from me today on Conan the Bacterium, Deinococcus radiodurans. I hope you liked the video. If you did, consider leaving a like. And if you want to see more content like this, then hit the subscribe button and the notification bell. And I'll see you next time in Tiny Giants.